This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM.
This is TWiM, This Week in Microbiology, where today we are recording a very special episode from the campus of the United States Army's Medical Research Institute of Infectious Diseases, located in Frederick, Maryland. I'm Michael Schmidt from the Medical University of South Carolina, and joining us from USAMRIID to explore the unseen life on Earth is Dr. Michelle Swanson from the University of Michigan. Woo! Welcome, Michelle.
Thank you. It is such a privilege to be with you all.
And to her left is my colleague from Washington University in St. Louis, Dr. Petra Levin.
Thank you. It's really a pleasure and also an honor to be here. And thank you for taking the time to come to this.
Our other co-host from TWiM, Dr. Vincent Racaniello, sends his regrets that he won't be joining us for this episode of TWiM, where today we're going to learn how military and civilian scientists of USAMRIID are excelling at their mission to develop biodefense solutions to protect our nation. For our viewing and listening audience, since 1969, USAMRIID has provided leading-edge medical capabilities to deter and defend against current and emerging biological threat agents. The Institute is the only laboratory in the Department of Defense equipped to safely study highly hazardous microbes requiring maximum containment at biosafety level 4. If you're unfamiliar with the term biosafety level 4 or its associated protocols, I'll refer you to a video that our TWiM cousins produced a few years ago from the National Emerging Infectious Diseases Laboratory, or the NEEDLE, for episode 200 of TWiM, which if you're behind on your TWiM episodes, that was well over 11 years ago. So you still have some time to catch up. But that episode is really worth watching. The research conducted here at USAMRIID has led to and is leading to vaccines, drugs, diagnostics, and training programs that protect our warfighters as well as all of us, the civilians. The Institute's unique science and technology base serves not only to address current threats to our armed forces, but is an essential element in the medical response to any and all future biological threats that may confront not only our nation, but the world at large. Today, to help us appreciate the service and the science that USAMRIID is providing to the world, we are fortunate to have a few of its rock stars who are going to share with us some of what they've been doing to keep all of us safe. So I'm going to start with Colonel Norman Kreiselmeier. Welcome, Colonel.
Thank you.
You are the director of the Core Support Group here at USAMRIID, and we're going to ask you a few questions in a few moments, but I'm going to continue down the line of panelists. Following Colonel K is Dr. Chris Cody, whose science we're going to tear into today because he has brought with him a paper that's going to be published in Emerging Infectious Diseases in a few short weeks that I think our general audience is going to find absolutely fascinating. And following Dr. Chris is Dr. Kirsten Ricks, who is really probably got the coolest job here on base. She gets to know what's going around the world before anyone else. She's in charge of the emerging diagnostics, but I'm probably botching that, so we're going to let her explain that. And following her is Dr. Eric Nguyen, who's got some really cool toys that he has been using to figure out some really complex problems as the microbes infect our cells. In fact, he's using a three-dimensional model of a human lung cell to actually witness how an infection begins to take place. So without any further ado, we're going to start with Colonel K. Why don't you tell us a little bit about how you got where you're sitting here today, what drew you to this position, and obviously stayed in green for quite some time.
Yeah, almost 20 years now. Well, I went to vet school. You know, I loved dogs and cats when I was a kid. Decided vet school sounds good, so I graduated from the Ohio State University College of Vet Med in 1992. I did dog and cat practice for 8 years and kind of wanted to expand my horizons a bit. So I got into a postdoc program in Cleveland. And in 2000, that started in 2000. In 2001, somebody flew a plane into a building that didn't sit well with me, but I was in the middle of my program. And in 2004, when my obligation was complete, I went to the recruiter's office. And I said, hey man, look, I'm a 38-year-old veterinarian. I don't know if you have any use for me, but if I can provide something to the Army, I would love to serve my country. And after jumping through a lot of hoops and getting waivers because I was over the age limit, they took me on board and they sent me directly to here at USAMRIID based on my experiences already. So I came here and I was in the vet med division as a lab animal veterinarian and have stayed in the Army ever since. And this is my third time back to USAMRIID. After going to other different assignments, I've always come back here.
Well, thank you for your service. And why don't you tell us a little bit about your day-to-day, what you're doing presently here at USAMRIID? Because I know your contributions and your group's contributions are so highly valued by everyone in this room, and everyone listening should really appreciate what you and your group are doing for us all.
Right. Okay. So yeah, I'm the head of the Core Support Directorate. So this is the group that supports all the scientists in doing their projects. So underneath me, I have the Veterinary Medicine Division, which cares for all the animals and provides the training for everybody that uses animals, as well as providing technical support and surgical support to anybody that has projects that require animal use. Underneath the Vet Med Division, we also have the Aerobiology group. They perform all the aerosol exposures for any projects that are looking at aerosol delivery of agents or pathogens. It's a very advanced group. It's the only group probably worldwide that can do aerosol delivery to the level that they do it. Next in line is veterinary pathology. They perform all the post-mortem examinations of our animal models, to include slide production, reading of slides just on the standard H&E level, but also down to immunohistochemistry, where we can identify and localize proteins in the tissues, particularly of the agents, but also of the different cell types, as well as ISH, in situ hybridization, where we can localize nucleic acids, particularly of the agents, the viruses in particular. Next to that is the quality group. They perform all the quality assurance. Our ultimate goal in what we do at USAMRIID is to develop countermeasures that we submit to the FDA for FDA approval. Submitting those drug requests to the FDA requires a very stringent package. The requirements are very detailed. And so every single piece of data that a machine puts out, every word that anybody writes is reviewed by our quality team to make sure it's consistent, repeatable, and meets the formatting to be able to be delivered to the FDA for consideration to develop and approve a new drug. So quality comes under core. I also have the Division of Medicine, which has our training and education branch to it. And so after we do all our work here at USAMRIID and we develop countermeasures and we deliver that to the military in particular, that needs to be, utilizing those things needs to be trained to the healthcare providers as well as the first line responders being the soldiers. So our Division of Medicine provides multiple different courses to train our military and healthcare providers on the countermeasures that we've developed.
Yes, please.
So one of the things I found really interesting was this aerosol division. Can you explain in the context of the countermeasures why that is so important? Because I think our audience would be interested, but also it goes to your mission.
Sure. Primarily from a biowarfare standpoint, our adversaries, more back in the day, were developing biological weapons that could be delivered by aerosol. We've heard of weaponized powders. So the Soviet Union was developing aerosols to deliver biological agents that could be sprayed over the battlefield or over cities. And so these are delivered by aerosol, and we were trying to develop countermeasures against that. So we need to replicate that capability so we can test what it's doing in our animal models in order to defend against it. We don't do anything offensive. We do nothing to develop weaponized powders ourselves. It's all to develop defenses against those aerosols that we might be exposed to.
Right. I was really interested in that also because the aerosols, what I didn't appreciate is normally when you get an infectious disease, it comes in through a very particular route. But by aerosolizing, you change the route and that changes how the disease goes. So it also gives you another understanding of how the disease could work.
Right. And we can alter that route in the respiratory tree, which aerosols are delivered by, and have that powder agent aerosol land in the upper respiratory tract, in the nose, or further down in the airways, or all the way down into the deepest parts of the lungs, the alveoli, because each of those areas have different cell types, different tissues, different immunology, so we can stratify where we want to land that agent and see what the pathology is or the infection is at each of those levels.
That seems very powerful for something like bird flu or for the avian flu, which for us would have to go much farther down in our respiratory tract, but normally doesn't happen. So you can actually do some of these animal studies where you're...
Yeah, you can land it where you pretty much want
that's
to.
amazing.
I am so impressed hearing about how your love of, as a child, of cats and dogs have taken you on this path,
Yes.
and you've gained expertise that is valuable in so many unexpected ways. Can you reflect on that? Because I think too often we ask young kids, like, what do you want to be when you grow up? Or they're about to graduate high school or college. So what are you going to do now? Like, as though you only get to make one decision. Could you reflect on that and maybe share some advice that you'd have for youngsters out there, new graduates?
Yeah, when I was going through vet school, I had my core group of friends, and we all, talked about what we were going to do when we graduated. And I had 3 really close friends. They're all doing exactly what they wanted to do while we were in school. If they knew that I was in the Army, for one thing, and working in a high containment lab, they would be freaked out because it's not what we talked about at all. I wanted to be a veterinary surgeon. I wanted to, fix broken bones. But life is about opportunities. And coming to USAMRIID was an opportunity that I took advantage of to, make a bigger difference than to just one dog or cat. Our impact is not just to the military, not just to the nation, but worldwide. And I wanted to be a part of that. So, when opportunities are presented to you, you should take them.
Yeah. And the more skills and the more experience you have, kind of the broader platform and the more opportunities that come up. So we're never bored, right? You never know what new opportunity is coming.
Never had a boring day in the Army. In clinical practice, vaccines and fleas and diarrhea, it's just very rote. Never had a boring day.
So no fleas, no diarrhea in the Army.
No fleas. Well, sometimes fleas. If we want them, there'd be fleas. Yeah.
Do you still have pets? Do you have pets yourself?
I do. I'm a cat person.
You're a cat person.
Yeah.
So one last question, then we'll move on to Dr. Cody. If you had to tell someone one thing about USAMRIID that you'd like them to know, what would that one thing be?
Oh, boy.
I know it's a hard question, but I'm going to ask it of all of you so you guys get a head start.
I'm just enthralled with our mission. To me, when I got the opportunity to come here and just learn about this mission, I had no clue that any group did this kind of stuff. I just felt that what we do here is very impactful, but under the radar. Nobody knows that we do this work, but it is so significant, not only for the active things that we do, but for the deterrence it provides to adversaries, which is something that we don't often capture or realize, that when adversaries know that we exist and do these things, they're less likely to try to, release biological weapons upon us.
But I want to dispel the idea that the things you do here are secret. Not at all. They publish their work in really premier peer-reviewed journals. They share that to really advance science throughout the country, the world.
Yes, everything's published.
Yep.
We're all public. We go to all the meetings. We present all of our information.
Yep.
There's no top secret anything going on here.
And to disabuse our listeners, they have to write grants to people. So, you think you're going to go work for the deep pocket Uncle Sam? Not so. You still have to write those proposals, and they're scrutinized as rigorously as any and all of our proposals are scrutinized.
The entire audience is shaking their heads in agreement with that statement. So
nobody here is getting rich.
No,
working at USAMRIID. I bet every one of these people can go out into industry and do much better taking an industry job than they can working for USAMRIID. But everybody here is here because they believe in the mission as much as I do.
So the three of us were treated to a tour this morning to our listeners, and it was phenomenal. But I think one of the great takeaways I got is during COVID, when many of us got to be couch potatoes and work from home, all of these folks in this audience were working double time. They did not shut down USAMRIID. They were working.
And they shifted their mission to the COVID pandemic.
Yes. They actually shortened that pandemic for us all, I believe.
Yeah, there was a time when we thought all the roads would be closed and nobody could travel, and our commander at the time wrote everyone an MFR, memorandum for record, that we could keep with us in the vehicle that says we are essential personnel in combating COVID. We are allowed to drive to the laboratory. That's how the military thought of our mission at that time.
During COVID, you did test development and also treatment screening for therapeutics.
Yeah, our mission in general is always diagnose and detect and subsequently characterize the agent, develop the animal model, which is then used to develop
We'll
countermeasures.
hear about some examples from your colleagues.
Therapeutics or vaccines.
But like the things that you guys did were not only for the military, they're for
The general public.
the general public.
Right. Our capabilities were leveraged for public use.
So we're going to transition one down. We're going to introduce Dr. Chris Cody. So Chris, why don't you tell us a little bit about your journey and how you ended up in a blue shirt instead of a green one.
Sure, absolutely.
And what you've been doing since you've left graduate school once upon a time.
Yeah, awesome. Before I get into that, I did want to comment on Colonel K. He's a phenomenal veterinarian, a veterinary surgeon, a veterinary pathologist. He's been here 3 times, and he's an amazing military leader. And so those of us that have been here for decades, we appreciate when they come back because they have that knowledge of us, and he is just the epitome of a military leader. So I personally thank you, and I know everybody thanks you for coming back.
You're here.
He owes me a beer now. So I graduated with a bachelor's in biology from University of Miami in 1997. And then I went to grad school at University of South Florida College of Medicine in Tampa with a PhD in medical science with a specialization in molecular microbiology. So from Florida, kind of moving myself up. And then up here, I moved as part of the National Research Council Fellowship. And I was a postdoctoral fellow starting in 2002. I interviewed before September 11th, but I started after September 11th, so it was a different vibe here. The gates went up and were different. But even before that, the mission really resonated with me. In 2005, I transitioned to a civilian principal investigator, and I'm currently the deputy chief of the bacteriology division at RID, which is the best division, not just at RID, but on the planet. So, yeah.
I think after the tour today, I think I almost have to agree with you.
Perfect.
I mean, your people are phenomenal.
Thank you. So the first 10 years of my career was spent on anthrax, but then as the DoD missions shifted, we started looking at melioidosis, which we'll talk more about,
yes,
and also plague.
Okay.
So can you tell a little bit about the way that you came in through postdoc? And is that something that people should think about? Because that's, a lot of academic grad students are obviously mostly at academic institutions. But then when they're thinking about going on and if they're interested in government service, this is a great opportunity.
Yeah, it was a great opportunity. And we still use the National Research Council postdoctoral fellowship through the National Academies of Science as one of the 2 main mechanisms to bring postdocs here. And those of us with opportunities, we're posted on the National Academies NRC website with research opportunities, and our points of contact are there as well. We also use the ORISE mechanism to bring postdoctoral fellows here too, which is through the Oak Ridge Institute, through the Department of Energy.
And for those of you watching, we're going to drop those 2 links into the show notes. So if you're interested in opportunities here at USAMRIID, you can click on one of those links and find out more.
Perfect.
And Dr. Kirsten Ricks, a Tar Heel.
No.
Wolf
bag.
Oh, yeah, wolf bag. No.
So why don't you tell us a little bit about yourself?
Yeah, so I came to USAMRIID in 2015, so this year in December will be 9 years. It seems like it's been forever, but not really. I will fight Chris in that DSD is the best division.
Yes.
Diagnostics, but I did my bachelor's degree at NC State, so not UNC as a Tar Heel, but I am a Wolfpack, go Wolfpack. But I did my degree in chemistry, so I came from a chemist background. I had a really great high school chemistry teacher, so I knew I always wanted to do chemistry. I also liked research, so I figured I would go to graduate school. But I finished my bachelor's degree at NC State in chemistry in 2010. And then I went to Vanderbilt University in Nashville for my PhD, also in chemistry. But I kind of fell into the diagnostics world because the lab that I joined did a lot of low resource malaria diagnostics. So biomedical engineering wasn't quite growing / at its peak like it is now. And NC State's a heavy engineering school. So I got my first exposure to where chemistry and biomedical engineering intersect and biology intersect at Vanderbilt. And so I did a lot of work developing tools that we can take out into a far forward area, low resource country to do malaria diagnostics or make the current malaria diagnostics work better. And so that's kind of where I got my toe in the water, if you will, of what the diagnostic world was like and what travel was like and working with other countries and teaching and all of that comes along with doing diagnostic work. So in looking for a postdoc, I knew I didn't want to stay in academia because I just wanted to try something else. I knew I didn't want to stay in the academic world. I wanted to either do industry or government, but a postdoc in the biomedical engineering department at Vanderbilt told me about the NRC program. I had no idea that those programs existed. I grew up in North Carolina, did my graduate work in Tennessee. I think if you're from this area, sometimes people tend to know, but that information hadn't been spread out yet. So he said, why don't you look at NRC programs? So I looked, I saw positions at NRL, Navy Research Lab, and USAMRIID for similar vibe type things in terms of diagnostics, disease diagnostics. Interviewed at both, loved it here. Everyone's like, oh, you must have read The Hot Zone. I didn't read The Hot Zone. I did not know what USAMRIID was. But I just fell in love with the people here and the mission. But the people was really what drew me here because they were so passionate about the mission. So I started my postdoc in 2015 and I never left. It's been a good journey, long journey. And I'll add something with the COVID story too. It was tough being a postdoc during that time. One of my coworkers and I were working on our CVs the other day and we were like, what happened in 2020? So it was a really difficult time, but I think we like to say that we pivoted to the COVID mission, but we actually did both. We did COVID and we did everything else that we're required and paid and funded to do at the same time. So we really were double teaming all of our efforts.
I was serious when I was thanking you guys of your double time.
Yeah.
It was truly double time. You did both projects.
Yeah.
All projects.
All of them.
Yeah.
And not only the COVID pandemic, but right now we're seeing a lot of scary headlines about Mpox, and you and your team have been working at the forefront there. Could you share that?
Yeah, so I think the Institute is unique in that we've A, been around for a long time, B, a lot of these bugs that are emerging, if they're emerging and we know about them, that's great. We've already done work on the viruses and bacterial families that are coming out of these regions, but sometimes new things emerge and they look like the things that we've already worked on. So then we can at least say I have a general picture I have a grainy picture of what I'm painting. Now I'll get like a finer detail with the technologies that we use here, sequencing, animal modeling, all that stuff. So when monkeypox came about, luckily we had already done some research on monkeypox here. So we had a little bit of a baseline, but on top of that, we already had a lot of work in the orthopox family in general with Dr. Hooper. I think he was on TWiM a few months ago. So we kind of already knew what we were doing in that space. We just needed to kind of pivot specifically for monkeypox. And so we had a good leg up on the research field, at least when it comes to that. And so then we just did all the things that we needed to do to get specific for the clade that's circulating. And then now we're still facing monkeypox and potentially a different clade. So with all of the experts in the field here, that's kind of how we can pivot but still have the expertise underneath to kind of help us move along.
Yeah, in particular, a diagnostic assay that you want to make available. Can you tell us about that?
Yes. So in the Diagnostic Systems Division, obviously, as it states in the name, we make diagnostics. But there's two types of diagnostics that you can use. The research grade, RUO, diagnostics, research use only, or you can transition that data into a more well-characterized, well-validated assay for FDA use or for clinical use. And so you can do that in our diagnostics division through our quality groups to basically say, okay, we've done the research, we've had the building blocks developed to make either a nucleic acid-based detection test or a protein-based detection test. We know about those for the COVID. At the beginning, you swabbed your nose, sent it off to the lab. That was nucleic acid testing. Later on, you swabbed your nose, put it on a strip. That was protein testing. So we can do both of those in the diagnostics division, but we have pipelines where we take whatever pathogen it may be. So for monkeypox, a couple years ago, it was clade 2B. We designed the Legos that you need to build that nucleic acid test, as well as the tools that you need for that protein detection test. And then we run it through the paces. We make sure it picks up what it should pick up, make sure it doesn't pick up what it shouldn't pick up, make sure that it's reproducible by user, and then make sure that it picks up live agent, right? Live agent and clinical matrix. Once we have all that data, then we can start to put the packet together for approvals to be able to be used for clinical use.
So what was also really fascinating is you have lots of diagnostic tests that are close to what might emerge or what hasn't really emerged, so that if something becomes more like a pandemic, you can send it to you're ready to go. You essentially have a collection of ready to go almost.
Yeah,
things.
absolutely. It's really important to be pre-positioned and ready. Like I don't have kids, but right, you're going to have your kid. You've got your go bag ready to go to the hospital. It's kind of like that analogy, right? Like something might come down the pike.
Readiness.
Yeah, readiness, right?
Military.
You just want to be ready. So, and luckily a lot of the things that we stay ready for are the things that our community cares about in the biodefense community. And so a lot of that is driven by leadership, by guidance documents, but a lot of it's also driven by our overseas collaborators, people that we work with. This is what they're seeing on the ground in their populations. We know that the Joint Force, we know that warfighters are there. And so we can also use that information to say, hey, Chem Biodefense Program, like, are you thinking about this? So it's a back and forth of getting information from the requirements documents and from people that are way above my pay grade, but also working in the international community and in the Joint Force community to understand, okay, this is what we need to be prepared for.
Right. And I think our audience will also be really interested to know, because you think about monkeypox, right? That can infectious diseases in general can infect anybody, right? And so why would you be working on monkeypox? What's the rationale for working on it here at USAMRIID?
So, I mean, historically, I think it's fallen in the orthopox viruses in general have fallen historically in that biothreat space. But monkeypox has emerged in both ways from a public health perspective, because we are a global community. We saw that with COVID. I think there were all of those maps of flights daily, and you're imagining, okay, there's people on these flights, they're bringing COVID with them. Monkeypox could be a scenario like that as well, in terms of if it can be spread through contact, through fomites, things like that. But I think as we start to think about the global spread and the global distribution of these things, that's where we start to get into the mindset of, okay, sure, it was endemic in maybe Western Central Africa. We've known about this for years, but the world is a very mobile community. People are spreading. The risk is just inherently increasing. And so that's why we're becoming more prepared for those types of things.
Right. So it's health of the warfighter,
right,
but also health of people, anybody who comes in contact.
Right.
Because it is infectious disease.
Yes.
So as our planet is warming and the things that have been put into the deep freeze are thawing out, a lot of popular science fiction, scary things always are talking about the bug that emerged from the permafrost that thawed. So are people with the crystal balls worried about the permafrost thawing?
I was at a joint senior leaders course the other week, and someone actually brought this up. So were you listening in?
No.
No, I would say that yes, the general scientific community is concerned. You see articles about it all the time. Again, to echo that preparedness standpoint, warfighters are all over the world. They're in all environments, hot, cold. So we just need to be prepared for, we don't know where the next fight is going to be. We don't know what our warfighters need to be prepared for. So in our minds, we are preparing just in case if there's an Arctic event. So we actually sent some team members from the Diagnostics division to Alaska to an Arctic Edge exercise. We call them exercises, where we basically tested out some of our diagnostic tools in the cold. Obviously, liquid freezes. We all know that. But how would you mitigate that? And how would you mitigate that in a low resource environment, right? I think something that we didn't think about was that if a thermocycler, so something that cycles the temperature for nucleic acid testing, if that gets cold, is it going to work?
It stays cold.
It stays cold. Sometimes the battery just says, no, I don't like this. I'm cold. I'm just going to stay and hibernate. Or it actually slows down your reaction because it takes longer to get to temperature and then it takes longer to cool. And so these are things that I guess you wouldn't have thought about just working in our lab or say going into tropical West Africa, working in the field. So from the standpoint of the permafrost, I can't comment on that in terms of what the crystal ball says, but at least from a preparedness standpoint, from what tools do we have in our toolkit, we're at least thinking about some of those things because we do need to be prepared and come up with strategies to basically figure out, okay, what do we need to design to help this liquid not freeze that's already in a pack? Is it a hot hands? Will that work? Can we come up with a device that doesn't need a thermocycler? Things like that. So that's how we're kind of approaching that extreme environment perspective, right?
Very, very cool. So that takes us to the last person on our panel today, Dr. Eric Nguyen, who is a contractor here at USAMRIID. So, Dr. Nguyen, why don't you tell us, or Eric, how you came to USAMRIID and describe a couple of the cool things that you're doing that absolutely fascinated all of us on the tour this morning.
Thank you very much. So yes, I am a contractor working with Geneva Technical and Professional Services. I joined in 2023, and well, let's actually start from like way sooner. So in high school, I basically got into biomedical engineering because I knew some people who were suffering from genetic and genital diseases, which in my mind was a fundamental injustice that I wanted to fight against. So I wanted to use my skills to basically help improve those people's lives and start fighting those diseases as well. But I didn't actually want to become a doctor. So biomedical engineering seemed like a good fit for me. I did my undergrad at Bucknell University in biomedical engineering and went in 2009 to get my PhD in biomedical engineering as well as at the University of Wisconsin-Madison. Studying with Dr. Bill Murphy in his bio-inspired materials lab. How many names am I allowed to drop doing this?
You can do as many as you like.
Awesome.
Karen's got a big recording card.
Awesome. Okay, so essentially my PhD and my postdoc were all about specifically defining biomaterials to work with either human stem cells or human primary cells. In order to basically get them to do what we need them to do. So basically decorate a polymer with a certain peptide or other molecule that can link to integrins or other signaling mechanisms of the cell, get them to form blood vessels, get them to form other tissues or organs, or start up a cell signaling cascade that is exactly what we need for something like a drug screening situation or a viral infection situation, et cetera.
And if I could interrupt, many people have probably heard about 3D printing. You can make a plastic object of various shapes. They're using 3D printing, but with different cells as the ink and giving them the right growth factors and creating little organoids that you could then do your experiments on, right?
That's correct.
It's fantastic use of your engineering and medical mission.
Mm-hmm. Yeah, so that was actually the point of my second postdoc, actually, which was to go from, I define the materials to let somebody else define the materials, let someone else provide the cells, and put them together in precisely defined formulations, different, extracellular matrix proteins, different cell types, and print them into defined geometric shapes to define tissues. In that situation, it was actually the human choroid and retina, and used those as drug validation systems. Well, it turns out at NIH, which I was doing my postdoc at, has a time limit for how long you can postdoc there, and I needed another position. And as a lot of different opportunities and doors were closing, I get a message on LinkedIn. Shouts to LinkedIn. I actually don't really use it unless I'm actively looking for a job at the time. But somebody sent me a message saying, hey, we think that your skill set is really appropriate for you to work at USAMRIID. Here's the project, here are the skills we want from you, here's who the client is, and all this really useful information. I back up the information with a job announcement found elsewhere, and I'm like, wow, this is actually not a scam. Let's look at this and actually work really hard on applying. But what's a USAMRIID? I didn't know what it was until we got there. But the big risk was that here's this person with a lot of biomedical engineering expertise with imaging and computer programming languages and other soft skills, but without any actual experience with virology or microbiology, anything like that. But it ended up being a good fit because I am adaptable. I can basically hone my skills to different projects, and I'm surrounded by the foremost experts in virology and microbiology that there are out there. So it ended up being a situation where I could actually work on a lot of projects I was interested but didn't have an opportunity to work on before. Back in my previous postdocs, the pandemic happened while I was working at NIH. And there were some efforts to try to pivot some of our work to things like trying to validate different possible anti-COVID compounds from a safety and efficacy standpoint. But a lot of different labs are really limited in their ability to really attack COVID and try to solve the problem here. So now I'm in a unique position where I can actually serve my country and be able to really try to make sure that when the next pandemic, like say a bug comes out from Arctic thaw, the catastrophic impact of that will be lessened, and I get to be there to try to stop it.
And I should point out, we were privileged, and we got a great introduction to biosafety here at USAMRIID. And you weren't just dropped into the deep end of the pool and said, here's Ebola, play with it. You had to go through rigorous training to work even at biosafety level 2. They just didn't throw you to the wolves, so to speak.
Yes, I did have to train, and I did have to make sure that anything that I touch was safely deactivated.
So actually, this seems like a good time to explain what the different biosafety levels are, because the audience might not all understand. So your lab is a BSL-2, right?
Correct.
Most of your work is, so that's things that can make you sick, but usually not. I don't know can you actually, maybe, Colonel K?
BSL-2 are typical endemic diseases which we might get exposed to on a daily basis. BSL-3 agents are high consequence pathogens that have high morbidity, high mortality, and transmissibility, but that we have countermeasures against, whether it's a vaccine or a therapeutic. So we can work with those at a BSL-3 level.
So tuberculosis would be a BSL-3.
Yes, TB is 3.
It's aerosol.
It's not one that we use.
No, it's not one you use. I'm just for people know.
For example, anthrax. We have
Anthrax.
vaccines and antibiotics against anthrax, so we can use that at BSL-3. BSL-4 is our maximum containment level. Those are our highest consequence pathogens for which there are no vaccines, therapeutics, or countermeasures against. So we work at those at the maximum level of containment.
So then they would need to be decontaminated so that you can examine the outcome of the experiment. The experiments are done at BSL-4,
Mm-hmm.
Dr. Nguyen, and
Yes.
and then essentially inactivated, not decontaminated, inactivated and given to you to do the analysis.
Correct. Yep. So a lot of what I'm doing then is there are two sort of main knowledge gaps that exist within our current drug discovery pipeline. The first one is the classic, okay, say that you've got a lead drug candidate that came out of a cell-based assay, either from a high-throughput screen or a dose-response screen. You're pretty confident that you can take those drugs into animals and get something that could work in a human clinical situation, and suddenly everything falls apart. Like something that worked in cells does not work in an animal level, and you don't really know why. The second gap is, okay, if we have a novel pandemic level drug or a novel viral threat coming at our warfighters, if we don't even know what it is, how do we even start analyzing it? How do we even know it's there, and what can we actually get a countermeasure done on time? So the use of 3D bioprinted tissues, microphysiological systems, or organoids are kind of we're putting in a lot of effort to be able to use that as a tool for the first knowledge gap. Essentially, can we go from cells to different cells interacting with each other in a three-dimensional environment, something that is more physiologically relevant to what would happen in a human, and see if any interactions there can actually explain why this drug does or doesn't work? Or can it actually mimic a more physiological system where a virus can attack human cells or human tissues in a more realistic way, and can the drug stop that? If we can do that, then we basically send fewer drugs to mice or non-human primates and save a lot of money being more confident in the drugs that we use. And in the second knowledge gap, we're actually leading on AI machine learning to be able to scan cells at a level that human eyes either can't look at 10,000 different images and make a judgment call on what's going on, or be able to identify a lot of different changes in a cell image that humans can't keep track of, and be able to link that to a gene expression regulatory pathway or another cell biomechanism, to be able to tell what the virus is actually doing and what drugs can we use to actually stop that.
So this gives you the opportunity to not just test one drug against one cell, but actually set up kind of high throughput screens and then use the computational biology to identify what's worth pursuing and
Correct.
verifying.
Yes.
It's great.
And that was one of the cool things from our tour this morning is you shared with us that you had just analyzed your data, where I think you probably looked at like 10,000 confocal images of an infection. Now, for the graduate students listening, he looked at 10,000 confocal images before lunch. I mean, this is where the science is going. And you guys are at the cutting edge with your instruments and your compute power. Now, I understand from our tour, it's one of your limitations was, where do you put all the images? I mean, you got 10,000 confocal images before lunch from one experiment. I mean, this is a good problem to have, but I mean, you guys are really pushing the envelope.
Yes, thanks and probably condolences to our information management division. They had to set up a very robust information handling and local area network to be able to handle petabytes of data. Not only from an image storage standpoint, but the microbiology division also produces a copious amount of gene expression, like RNA-seq data that needs to be stored as well and cataloged. We couldn't do our work without them. So.
Right, because you're getting data not only on how the pathogen is responding to your potential therapeutic, but also how the cells are responding.
Yep.
And you can essentially get counts. You can get so many pieces of information out of one image because you're using 3 different fluorophores. And yeah, it's incredible. So how much is a petabyte? What was?
We know what a gigabyte is because we have jump drives.
I think it's 1,000 terabytes.
Yes.
Yeah.
Yeah, it went from gigabyte to terabyte. And now petabyte.
And is that
is the next.
an increase of 3 with each of those leaps?
Yes.
So one run of your, like when you're testing on that amazing microscope in a box that can do thousands of images, one experiment, how much data is generated?
Okay, so we're not going for the entire petabyte just yet.
Okay.
One experiment is going to be about 10 gigabytes, but we're having to run hundreds of experiments for one project, and we have multiple projects going on. Depending on complexity, if we try to start breaking into 3-dimensional imaging of a bioprinted tissue or an organoid, that'll ramp up considerably. If we need to take separate sets of images for a machine learning experiment and be able to store the training data on there, that will also start ratcheting up our data use considerably. So we're spread out in a lot of different directions, and that's where it all adds up.
So do you have data scientists working with you to analyze this or sort it, or even with all the genomic and RNA-seq, all the data that's out there here, because we were also with the sequencing data, how is that all being managed?
Yes, we do have dedicated data scientists. Raina Kumar is actually in the audience sitting with us right now. We've worked closely with her with the gene data analysis. For a lot of the AI machine learning, we actually use a proprietary machine learning software suite that comes with our Opera Phoenix imaging systems. And I'm mainly heading the charge for that to not only try to teach the machine learning models how to dissect our data, but also how to check the quality of the data outputs and see if it's actually answering the questions that we needed to answer.
So actually, I have another question, which goes to that you have the data science resource. So for all 3 of you, what's incredible also here is that there's so many resources, both intellectual from personnel, but also equipment and things that you can use.
Core facilities.
I don't know whether that, if you come from, all of you were in academia at some point, including yourself. But is that something, how do you make sure to build? How do you decide what you need and build that here? Or
we try to be a one-stop shop with what we do.
and the bugs we work on, a one-stop shop is preferable. But that is very much a challenge because as the technology improves, the cost goes up, the cost of maintenance goes up, the data scientists needed to make the data make sense, that all that cost goes up. So balancing growth in the right direction and keeping a balanced budget can be challenging for sure. But we're so proud of the technology that we have here and the capabilities that we have here, and I think we're really as close to a one-stop shop as I think we could get.
Yeah, one-stop shop is a great description of what we saw today in the tour.
And the synergy between all the teams. Everyone talks to everyone. So my one question then is, how do you guys get anything done? Because you're all doing such cool things. It would, you go get a cup of coffee and just finding out, well, what did you learn last night? And the tour was so full of information. You guys just must go home like blown away every day.
Well, there's a lot of communication.
Curses, ladies.
Well, I think honestly, like the people in this room, but even the people that aren't in this room, as Colonel Pitney said, other people have said today, there's a little over 600 people that work here. It's because of everyone that we can do what we do. Like personally, I have technical staff that are in my group that have been here almost as long as I've been alive. So and they're just dedicated to this place, and they're dedicated to what we do, and they really take their job seriously. And I know that I can rely on them. So I think that helps us sleep at night and also just get the work done, because we know that we have such a fantastic team here to do it with us. It's not just us. It's not just the people in the offices. It's everyone that works in the building.
Yeah, teamwork is key.
Yeah.
For the breadth of mission that we have and the limited resources, if we don't work as a cohesive team, we're not going to get anywhere near where we need to be. And Kirsten's division and Betsy, we work together so closely. We don't try to reinvent the wheel. We don't need to. They've already done that. Animal modeling is not diagnostic strength. So when they need help, they immediately come to us, and we sometimes within 10 minutes, we'll have a collaboration sketched out. So when the whole institute really functions that way.
And I think that was what another impressive piece of the tour here today offered us, is that animals are used almost as a last resort. You really want to use cell culture to get the clues before you have to put these agents into the animals to effectively maximize the animal resource that you have, and don't waste the precious animals.
Yeah, it's a huge push right now across DoD to look at organoids, organs on a chip, and for USAMRIID in the coming years. I mean, we're going to have a huge footprint in the technology, and for some of us old timers, it's a little challenging to get used to the new technology and trying to extrapolate the complexities of a whole animal model to a few chips. But the technology that's there now is really impressive, and we're looking forward to trying to apply that to our work.
Well, we promised the audience that we'd get to some science here today. We could talk to you.
Additional science.
We love hearing all of these stories. And Dr. Chris Cody was one of the authors of the paper that we're going to discuss. Characterization of the virulence of the Burkholderia pseudomallei ATS 2021 strain unintentionally imported into the United States in aromatherapy. Now, I can tell you that aromatherapy units are the plague of our infection control team in my hospital. The infection control team has a mission every Monday to ferret out all the aromatherapy dispensers that have been bought at Target and brought in over the weekend secretly, because they're always diffusing, and they literally go around with their nose looking for the culprits. So why don't you take us through, or at least introduce us to your paper of how Burkholderia level 3 agent got into something as innocuous as aromatherapy that I think everyone has at least experienced once in their life.
Yeah, it's a really interesting time to be working on Burkholderia pseudomallei. So it is in a different context. It's recently been declared endemic to the Gulf Coast of Mississippi. So we know that it is endemic to the continental United States. That was just discovered in the last year or two. This incident that you're speaking about in 2021, there were 4 individuals that became sick that were later diagnosed with melioidosis in 4 different states. It was Minnesota, Kansas, Texas, and Georgia. And it was really a masterclass in CDC investigation. Here they had 4 individuals that had no history of international travel, and they all had melioidosis. So CDC had to figure out where that was coming from.
So what is melioidosis for the uninitiated in our audience?
Sure. So melioidosis is a disease caused by Burkholderia pseudomallei. And historically, it was always studied in Southeast Asia and Northern Australia because those are kind of your hyper-endemic areas. It's always been known to be in that tropical, subtropical area, but over the last decade or so, the characterization and identification has been dramatically improved. And so we know that it's in Africa, it's underreported in Africa. India is a big hotspot right now. And then it's in South America, the Caribbean, and it was just recently identified in the Gulf Coast of Mississippi.
And tell us about some of the presentation of this disease and some of the scary neurological sequelae.
So it's a very, very challenging bacterium to work on because it is so insanely diverse in clinical presentations. And that's in humans and also animals. So this bacterium can infect almost every animal known, from marine mammals to humans to raccoons. For an example of this individual that got sick in Texas, it was a young child that had neurological manifestations. That family actually had a pet raccoon that also was exposed to the aromatherapy, and it was identified that this raccoon also had melioidosis, demonstrated neurological signs, then died shortly after. So the clinical presentations can be everything from flu-like to a much more acute pneumonia. And this is the severe disease is mainly associated with other risk factors such as diabetes or alcohol abuse, things of that nature. It's also very tough to model because it's known that it can infect a healthy individual and kind of hang out in your body for decades until you become immunocompromised. And so there are examples from World War II of warfighters being exposed in their teens in theater and showing never going back to that part of the country, part of the globe, and then developing melioidosis decades later. And so it's been on the DoD mind since Vietnam as well, because the area in Vietnam is prime Burkholderia pseudomallei location. They love that environment of the rice paddy of that tropical environment. So they were worried that with all the helicopter wash, that we would have a lot more warfighters that had been exposed to Burkholderia pseudomallei. Fortunately, that hasn't come to fruition. We didn't see a huge number of Vietnam vets that did become infected. So it's just a really interesting and challenging bacterium to work on that is not really known by a lot of people. And so when the CDC had these 4 cases of individuals that had never left the country coming down with melioidosis, they had to really work hard to figure it out. And it all came down to a Better Homes and Garden aromatherapy spray that was imported from India. And it was thought that this product was made with contaminated water at the source.
So was it surprising that it was only 4?
So we've been to meetings that, this was sold at Walmart across the country. I've been to meetings where some individuals hypothesize that there are still likely hundreds, if not thousands, of bottles of this spray out there. But we have only had 4 documented cases. There could have been other people that got ill and were just misdiagnosed or undiagnosed because melioidosis is so challenging to diagnose. It can mimic other things such as tuberculosis. But it is a really, really interesting case study.
And so the bacteria is able to survive I don't even know what they use for aromatherapy, some kind of alcohol-based or...
Yeah. So that product has been discontinued. So
thank you.
Thank you, Walmart.
Yeah. So, and as a side note, we did take a field trip to Walmart and buy other Better Homes and Gardens products, similar scents. And we did try to spike it with our bacteria. And the formulation that is sold at Walmart kills the bacteria within seconds to minutes. So what happened with that lot of manufacture, it's unclear, and I'm not sure we'll ever really figure it out.
Because you were never able to recover the original aromatherapy material.
Well, the CDC did.
Oh, the CDC
Yeah, they
did.
were able to isolate it from those bottles in some of these cases.
And the strain was kind of a super strain in a number of ways. Phenotypically, you characterized it. Could you tell us more about that?
Yeah, so it's called ATS 2021 because ATS stands for aromatherapy spray, and that's what the CDC named it. It is from India, and we don't have many Indian strains, so it gave us the opportunity to look at an Indian isolate. And out of the 4 individuals that were ill, 2 of them had notable neurological melioidosis, and 1 of them had some cognitive issues. So at least 50% of the individuals had neurological melioidosis. And so we know that strains of B.pseudomallei in Northern Australia and India have a propensity to have a neurological tropism. And so this strain also gave us the chance to start to look at that. As far as virulence, it matched some of our most virulent strains that we have from Thailand and Northern Australia. So that was important to document. It has also some really interesting biofilm properties. And so this was work done by Joel Bazoo and Kevin Malinick. And they documented that this strain can form really, really robust biofilm at 37 degrees.
Body temperature.
Body temperature, exactly. So there was a previous paper from Thailand that said that they saw no correlation between biofilm production and virulence when the strains originate from Thailand, and virulence is measured by the intraperitoneal route of a mouse infection. We did the experiment.
Intraperitoneal, explain that.
Intraperitoneal, so that's an injection into the gut, basically, into the peritoneum. And so it's a really simple, nice way to do infection that every lab can do. You don't need anything fancy. When we did our experiments using small particle aerosols, and we correlated it with the help of our very amazing biostats division, we had a correlation that did show that high biofilm formation did correlate with virulence through that route. And so our data corroborated the Thai paper, and that we didn't see that correlation with the intraperitoneal route, but we do see it in the small particle aerosol. And looking at pathology samples, the way that this bacterium just coats your nasal passage and your nasal turbinates, it sort of makes sense that if this bug has a high propensity for biofilm formation, it's going to set up a nest, for lack of a better term, and kind of just hang out.
And this is sounds like, especially from an aromatherapy spray, the more normal route to be infected with this organism that you're very rarely going to have it injected into your abdomen accidentally.
Exactly. And everybody in America knows what a nasal turbinate is because we all had the brain biopsy with the COVID swab. That's where it was going, the turbinate.
I just learned that term.
Right.
And the challenge with organisms that form biofilms is that unlike a free-floating bacterium or virus, they can be eaten by our white blood cells. But when you're in a community and they have a thick matrix over it, kind of a shell, goopy shell, now not only can't you get white blood cells in to eat them, but also antibiotics don't cross that biofilm gamish. So hard to clear, pretty tenacious.
So you were infecting the mice using your aerosolizing equipment here?
Exactly.
Yeah,
so that's where that
cool chambers. Yeah. That's great. Wow. You also found that it affected oligodendrocytes, and that might connect with the neurological pathology?
Yeah, so we're a little behind the curve with neurological melioidosis here at RID. We've only been looking at it for about 2 or 3 years, but it's a really poorly understood disease manifestation. And so what we had done in a serial time course, we had sampled the brain of the mice, and then we worked with Kirsten's division to do some host transcriptomics and start to look to see what genes are upregulated or downregulated in brains infected with this bacterium. And it seems that oligodendrocytes are at least one population that's very impacted by this infection, and not just very impacted, but impacted very quickly an infection.
Tell us more what those cells do.
So oligodendrocytes are important for myelination. And so they're going to wrap your your neurons in myelin and allow for good transit of electrical signals. So similar to wiring in your home. And so when the oligodendrocytes populations decrease or they're not functional, that myelination is broken down. And demyelinating diseases such as multiple sclerosis and things like that, that gives you an example of how important myelination is. Interesting to us was some of the clinical signs that our mice were showing with this strain really were mimicking something that could be perceived as a demyelinating disease. They would appear uncoordinated. They would have trouble with movement, and they would also have extreme sensitivity to touch. So when we would do clinical examinations, the mice would just react very suddenly and very strongly because we believe of the manifestations of the neurological melioidosis.
So this disease crosses the blood-brain barrier. This is the big problem with this particular strain or group of strains. Is that the...
Yep. So some of the strains they can get into the brain, and some of them have an allele that allows them to move more freely in the brain, I should say. The one caveat of our work is that to date it's been done in mice, and the anatomy of a mouse is obviously much different than a human or non-human primate. And so our work is early in that regards, and so we need to make sure that what we're seeing in mice can also be translated to larger mammals with different anatomy.
So I think in the paper you say that this protein, or BIM-A. So BIM-A, BM allele of this protein is sort of variant of this protein, which is important. So this bacteria can move between cells,
yep,
and possibly across the blood-brain barrier by polymerizing actin behind it. So it's in the same family as like Listeria, not family, but
Shigella.
Same mode of action,
exactly,
or Shigella. But do you think that ability to move between cells is what's allowing this particular variant is particularly good at it, that's what's allowing it to cross blood-brain barrier, kind of like Listeria might?
So I don't want to hypothesize if it's the actual crossing of the blood-brain barrier, but once it is into the brain, I think it reacts to that brain environment very differently with the allele. And so most of the neurological melioidosis strains have the BIM-A allele that is more characteristic of Burkholderia mallei, and that's why it'll be called BIM-A, BM.
Okay.
Most of the common lab strains from Thailand and other parts of Southeast Asia, they have what they call the BP allele, so that's BIM-A, BP, and that one is less associated with neurological melioidosis. So there's been a lot of work in Northern Australia and Southeast Asia to try to figure out this mechanism of action, and we're definitely late coming to the game. But this strain has given us kind of a leg up and a really good tool to begin to kind of tease that out.
Do you think that's the major variance determinant that's differentiating between the different strain families?
It's certainly a major one.
So, Dr. Ricks, when Dr. Cody came to you with this problem, how did you go about dealing with it? I mean, it's not an off-the-shelf traditional clinical diagnostic type of test.
Right. So again, the theme today, preparedness, right? Preparedness. We know that these are pathogens, these are targets that we need to be prepared for. So from my protein hat side of the house, or just immunodiagnostic side of the house, when he approached us, luckily we had already been introduced, again, this interconnectedness too, we had already been introduced to one of his collaborators, Dr. Alcoin from the University of Nevada, Reno, right? Yes. They had been collaborating with them for a long time on antibodies to look at them from a therapeutic perspective. But often those antibodies from a therapeutic perspective, they can be used for a diagnostic as well. There's nothing out there saying nope, these are only therapeutics, these are only diagnostics. They do work for both, at least from a research perspective. So we actually had already started working on a Burkholderia detection assay for the capsular polysaccharide on the surface. Just preparedness, have it on the shelf just in case you need it. And then from the transcriptomic side of the house that you guys were talking about earlier, again, it's one of those things that our funders are really interested in because there's this whole movement towards you have the host, our bodies, and then you have the pathogen, the bug. we want to understand, what does your body say and do when it's infected? And then what does the pathogen look like when you're infected? What does that bug do and look like when you're infected? The closer that you can get to time zero of infection, you're going to get that information from the host biomarkers. So our group has been funded for this type of work for several years now. We can't do that absent our friends in bacteriology and virology and their animal modeling samples. So we've been working together for a long time already. So when this strain popped up, he said, "Hey, do you guys want to work together?" And we were like, no, we're ready. Like, let us know what you need. We'll help you. We have whatever you need to support you. So strain came in, they characterized it, animal study was run, samples were transferred to us, and then we did the work that we needed to do to support them in their study. And then it's awesome that it turned out into a paper, and it's a really interesting story. I think the nice thing about a lot of the work that we do here is that they do tell stories. At the end of the day, science is storytelling as well, and who that story gets messaged to. So I thought this was one of the more interesting stories that I've looked at for sure in my time at USAMRIID.
Well, as I said in the beginning, aromatherapy is something that everyone has experienced. I mean, to worry about the thing that you pick up at the store, bring home and plug into your wall, and it could be making you sick, is really something that we need to think about. And unlike the food we eat, there's no regulatory effort on aromatherapy, at least to the best of my knowledge. Is there?
Not that I'm aware of.
Is there a therapeutic against Mallei?
So B.pseudomallei is often intrinsically antibiotic resistant, but there is, most of this work has been from Northern Australia, Darwin, and there's very good papers describing the therapeutics, but it's months and months and months of antibiotics, often IV for months, and then often oral eradication for months as well.
Because it's intracellular or maybe in biofilms.
Exactly.
It's also hard to diagnose, so you have to recognize it. Yeah,
in those hyper-endemic areas, those guys know what they're looking for. But I'll tell you, in Kansas and Minnesota and Gulf Coast Mississippi, they weren't necessarily looking for it.
And the other aspect is that, again, these are biofilm-based infections initially, and they don't really respond all that well to antibiotics because they're simply not growing. And antibiotics are designed for actively growing cells.
Yeah.
It's a rare number that, work against the ones that are just sitting there.
Yeah. And Matt Meinig in our group, who's our chief of bacterial core therapeutics, he's really interested in not just that, but also as the bugs get into the brain, is that sort of a privileged site that's it has a strange interplay with some antibiotics. So we're starting to ask those exact questions.
And you don't necessarily want a strong immune reaction in your brain because in addition to killing the pathogen, it'll take out the brain.
Exactly.
So that's a bad day for all.
I will say I am comforted that you didn't find any in the samples at Walmart or were unable to grow in the
I can
current
tell
formulation.
You the new Better Homes and Garden, pretty good.
Yeah. That's important. Don't want to scare anybody.
Don't want to scare anybody.
I'm sure I speak for many of our listeners when I say I can't tell you how much we admire and appreciate all the work that you all are doing to advance public health and for the greater good. So thank you all very much.
Thank you. Thank you, guys, too.
Yeah, thank you in the audience. So on behalf of my co-hosts, Dr. Petra Levin and Dr. Michelle Swanson, and our dear friend and absentee, Vince Racaniello, I'd like to thank all the individuals who were dragooned, is the right word, to the panel here this afternoon at USAMRIID, and everyone who made this visit at USAMRIID possible. All the people on the tour this morning that really gave us a phenomenal overview of what you all are doing to help keep us all safe. I want to personally thank you for all that you're doing. And also thanks are owed for inviting us and sharing with our audience the important work you all are accomplishing on behalf, again, of keeping us all safe. And finally, a special thanks to my point of contact for the visit here today, Lieutenant Colonel Kelly Hurahan, who helped us all navigate the complexity of visiting this remarkable campus. So on behalf of the TWiM team and Microbe TV, thanks everyone for attending, and thanks you all for listening.